Okay, great. So thank you very much for the invitation. It's a great chance to present our work and also share with you some of the progress that we've been making getting this small company started. I want to start by asking you just quick question. And if you can answer this, I think you could have developed the technology that I have developed. So what you see here is a protein that's bound to a DNA. It's a double helix DNA. And this could be a ligase, it could be a regulatory enzyme, but these are enzymes that recognizes specific molecules on a DNA, which is kind of have a very rigid structure, and it binds to it. The question is, if this was not double helix, if it was a single helix, what would be the molecule that would specifically bind to this single strand of DNA? Yeah, so it's actually not it's not a protein. It would be another strand of DNA that has a complementary sequence. So we're really not working with the DNA, but we're working with a protein that falls into triple helix. The idea of forming the secondary protein structure rather than tertiary protein structure interaction as a way to target or bind molecule kind of has been the basis of our technology. So we're all familiar with collagen. It's the most abundant protein in our body. And you can think of this as the structures that you see in this when you're building a house. Now, what happens when you have a damage, when the tree falls on your house? Of course, you have to fix it and build it. But before you build it, what do? You actually have to first get rid of the debris. We have to demolish. We actually have to degrade the house quite a lot before one can build. So that happens same in the case of our tissues, and the same case for the collagen, which is the structure of the house. Especially the disease that are known to be degenerative, such as arthritis, cancer, and an early stage of fibrosis. These degraded collagens are very, very common. And what we've been trying to do is to target these degraded collagen as a new type of a marker for detection of the disease. So if you look at the structure of collagen, the fundamental structure is triple helix. This is where the analogy becomes between DNA and collagen. When you have a disease and when you have an enzyme that starts to degrade collagen because of the healing process, what happens is that the collagen gets chopped up and the triple helix unravels into single strand. Now, rather than trying to detect or target these single strand collagen using an antibody or phage displays, a complicated process, we thought, similar to what I talked about in that first slide, if you can have another type of collagen-like molecule that wants to fold into triple helix, this is going to be similar to primer binding to DNA during the PCR process, that this molecule will stick and hybridize to this single-stranded collagen molecule by the formation of the triple helix. So we have here a relatively simple structure which is taken from the collagen. They are very hydrophilic, no hydrophobic residue, so they don't stick to anything else. It's almost like PEG in terms of their property. They don't stick to anything else, but it will only find and hybridize to denatured collagen. Because they are inert, they also have very high serum stability. That means you can use it for in vivo application. The problem is then you can conjugate these type of molecules, which could be a fluorescent probe, or it could be contrast agents, depends on what you want to use. And ultimately, you can even attach therapeutic moiety so that you can use this for drug delivery. So where have we used this? Well, it turns out that we can do a lot of things with this. Most obvious and most translational part of this has been that it can detect in a tissue section a disease because many of these diseases have denatured collagen in it. So we're talking about OA, arthritis, breast cancer, inflammation, and even early stage of heart attack where there's a remodeling activity of degrading of the collagen. And what's key here is that here, it's unlike the pathologist, they have to look at the histology and look at the overall morphology to determine whether you have a disease or not. We have a direct readout where the signal itself, the positive signal itself, will tell you whether you have the disease. And the level of the disease, how bad it is, also correlates with the intensity of these signal. So it will be very useful for example, if you want to automate the detection process. Not only that, we can also use this in vivo. I told you that the molecule is very high serum stability. Not many molecule works in glassware, works in vivo, but because the molecule is so inert and doesn't stick to other things, they will circulate the body and find a denatured collagen. So here you can see the detection of arthritic condition, Marfan disease, where there's high bone remodeling activity. And more recently, this is actually the work with Alana Alom, who just spoke, the data that shows that we can also detect the bone remodeling process as a result of metastatic breast cancer growth. So here we are using actually a dye that is fluorescent in the red color so that we can see through the animal. But we have actually more. So it turns out that the peptide can not only detect these degraded collagen that's a result of enzymatic activity. We can also detect collagen that are mechanically damaged. So why would this be interesting? Well, there are a lot of collagen in our body that serve as a mechanical purpose. Your tendon, right? Your ligament. You have a heart valve that's full of collagen. So what we did was we took a mouse tail tendon, stretched. This is the collaboration with Jeff Weiss in our department. And surprisingly, what we found out was that when you have a damage to your tendon, even very early on, there's really no sign of injury, your collagen molecule are actually partially unraveled. And your peptide, our peptide, is able to tell you that there is an injury. And what's really cool about this is that this opens up a whole new possibility of detecting early detection, especially for athletes, right? They want to know whether they have some condition that may ultimately lead to an injury. We can detect them very early. So the reason why we were able to have so many of these very interesting use of these compounds, I really think that is, and something that Glenn has mentioned in the first place, is the simplicity. The molecule is just very simple, composed of glycine, proline, hydroxyproline, very simple molecule, very hydrophilic. This is not an antibody. This is not a complex organic molecule that we really designed a lot to have them bind to something. We really took advantage of the nature, the fact that the nature gives us this very unique triple helical structure, and then the structure only exists in collagen is really what made us make this happen. Now, how did this all get started? Well, it didn't really start the way you may think that it started. The initial project that we got this going was actually to find a peptide that binds to collagen. And I'm showing you here is one of the very first figure that my R01 that we submitted several years ago. And this was done in order to functionalize collagen so that we can do tissue engineering. When we actually did the experiment, and we got completely opposite or devastating results. So this was a complete failure because the molecule did not bind to collagen, or that did not bind to intact collagen. It bound to denatured collagen. So normal people would have just said, okay, too bad, let's find another molecule that binds to the intact collagen. But we were lucky enough that we had a wonderful student who worked in my lab, who really turned this kind of unfortunate result into something that's really interesting and young. And we realized that, maybe targeting the denatured collagen would be much more interesting than targeting the native collagen because of all the reasons that I've mentioned here. Now, we made the discovery, and this was actually all happened at Johns Hopkins University, which I came from. It's been about 5 years, I think, since we've got the discovery. We published the paper, we filed patents, but nothing really happened. Like, I was busy writing Grad, Yang was busy getting his thesis defended, and our tech transfer office, unfortunately, was terrible. They just had no capability to do any type of market research. So we just kind of sat there doing nothing, but wanting to commercialize this. So that's why we came over to University of Utah. And of course, we have I came because of a wonderful collaborator, that's pharmaceutics, drug delivery, some of the people here that I can collaborate with. But really, I knew that the University of Utah had the top-level support for this kind of stuff. And of course, Yang, who was the inventor of this work, had the entrepreneurial mind that he wanted to. He really wanted to see this go to the market. So I was lucky, we were lucky in that sense. So after coming to University of Utah, we started talking with physicians, researchers, we did a lean canvas analysis, and we soon realized that our dream of using our peptide for any type of diagnostic or therapeutics really is far-reaching. It's like we were really not ready for that type of commitment, right? We need FDA approval, we have to go through so many different things, and we weren't really going to invest, we weren't able to attract any investment because that was still really far. However, we found out that there was a great interest in people who want to use our peptide as a research reagent and also as a staining for histology of biopsies. So that is the decision that we've made. So let's work on something easy. It doesn't require FDA approval. We don't really need a lot of capital investment for making this, and pursue that aspect. Now, another aspect of this, selling this as a research reagent, is that when we do that, we're actually having other people who actually buy our product to do the work for us. Well, these are the people who are going to purchase our probe and use it for various ways to detect collagen in whatever disease that they're working on. And we clearly saw that we can use their result, their data, for commercialization of our product and development of our future product. So that's what's happening right now. So we formed the 3Helix. We're kind of laying down all the basics that needs to be done. We have acquired some SBIR fund. We have some private fund. I think we have a little bit less than about $500,000 of money right now. Commercialization effort that we are going through. And at the same time, we're trying to develop next generation peptide, which is more user friendly. So we are actually at a very early stage. We're about to start marketing. So we'll see what happens, but I'm feeling really hopeful that good will turn out later. So I think with that, I will end my presentation. Thanks.